Hello, I will talk about glycoproteins and proteoglycans, the post-translationally modified proteins that bind. So I will have a quick overview of monosaccharides, modified sugars, oligosaccharides, and polysaccharides. And I will talk about two groups of modified proteins, glycoproteins and proteoglycans, their function, synthesis, and degradation. I found a couple of videos that you may find useful to understand what the proteoglycans and glycoproteins are and where they play a role in the human body. So this is a reminder when you learned about carbohydrates and carbohydrate metabolism in the previous block or previous blocks, you learned about monosaccharides and their use in energy generation, as well as you learned how they can be turned into glucose and how glucose can be stored in the form of glycogen. You also learned, related to that one, that the monomers can bind to each other since they have aldehyde or keto groups that are reactive, and how we can form oligosaccharides, shorter polymers or polysaccharides from these monomer sugars. Now, we will more focus on today on how we can use the carbohydrates to modify proteins. I will not talk about modified lipids, but glycoproteins and glycolipids and proteoglycans, two of them will be in the focus of my talk, are very important physiologically in the human body. So in this case, carbohydrates are covalently attached to the protein and they can attach through an amino acid which has an imino group or an amino acid which has a hydroxyl group in the terminal position of the side chain. So what you learned until now, that we eat carbohydrates, of course, the monosaccharides are absorbed, and these monosaccharides can be used for energy generation in every cell, majorly glucose or the others that are turned to glucose in glycolysis, and we can store that polyglucose away in the form of glycogen. You also learned a little about how sugars can be interconverted each other through the glycolytic and gluconeogenic pathway. You also learned a little about that we can modify these particular sugars. If you remember, you learned about uronic acid, which is an acid derivative of glucose. Now, we will talk about formation of more complex carbohydrate, not just glycogen, which is a polymer. And also I will talk about how the covalent modification of proteins with carbohydrates is going on. So what types of carbohydrates do we have? We have simple sugars, we have modified sugars, so derivatives of these simple sugars. We have polymers, and the polymers can be either the same simple sugar repeatedly, like glycogen, as a glucose polymer, or it can be a polymer of simple sugars and derivatives that build these more complex carbohydrates. Any of these can bind to proteins. So simple sugars, what you learned, glucose, galactose, mannose. Of course, we learned about fructose too, but it is better if we don't eat a lot of fructose, if you recall. Now we can modify these sugars, and this two figure shows you that we actually, in the first step, the hydroxyl group was changed to an amino group, and in the next step, the amino group was actually acetylated. So these two modified sugars, N-acetylglucosamine and N-acetylgalactosamine, are very frequent participants in the sugar units that are put on proteins, either in the form of glycoproteins or proteoglycans. Now, they can be polymerized. As I said, it can be the polymer of, that can be disaccharides, trisaccharides, tetrasaccharides, oligosaccharides, and polysaccharides. They can be the repeat of the same sugar, like maltose. You remember maltose was one of the disaccharides that was produced from glycogen in the digestive tract. It is composed of glucose. Or you also learned about heterodisaccharides, which one of them was lactose, glucose, and galactose components, and the other was table sugar, which was fructose and glucose. Now, these polymers, again, as I mentioned, can be made of the same sugars or can be made of different sugars and sugar derivatives. Please remember that these sugars, modified sugars, are dipeptide, disaccharides, trisaccharides, oligo or polysaccharides can bind covalently to proteins and lipids. And this is how we form these very complex molecules in the human body. This is for your information. Unfortunately, in this course, I don't have an opportunity to teach a little bit more cell biology. But if you are interested, I will really take only 1 minute of your time and I will not test you on this one. But everything which we do with the proteins, we modify them post-translationally. So you may recall that we have the 20 amino acids we activate. Of course, the proteins are encoded in the DNA. We activate and transcribe and then translate that particular message, and the ribosomes that participate in synthesizing the protein. Now, it depends what the protein is used for. It can go to two different pathways. One pathway is which is actually happening in free ribosomes, that cytosolic proteins will be synthesized, and also special proteins that have a tag on them will be synthesized, folded, and directed to the intracellular compartments, which can be mitochondria, can be peroxisome, lysosome, and so on. So these are the intracellular pathways. The other way of synthesizing proteins is actually with the with the implementation of further modifications in the rough endoplasmic reticulum. So ribosomes attach to the endoplasmic reticulum. They actually put the freshly synthesized nascent protein to the ER, and in the ER, a lot of different modifications will happen, including the attachment of carbohydrates to this nascent protein. Now, then it goes to the Golgi for further modifications, and these proteins, this pathway is actually called the secretory pathway. So what do we do with these proteins? One is lysosomal proteins, and the other is putting the proteins into the plasma membrane or secreting them out of the cell. So these glycoproteins and proteoglycans are going through the secretory pathway, and thus they will fill their role either in the plasma membrane or in the extracellular matrix. All right, so biosynthesis of complex carbohydrates are non-template, which means that we have the protein which is coming from the ribosomes to the endoplasmic reticulum, and there should be enzymes that will start to put together those glycosylation simple sugars or modified sugars or oligo or polysaccharides and attach it to the protein. Now, how do we regulate this? What will be put on certain proteins is upregulating the expression and activity of so-called glycosyl transferases. Every glycosidic bond, every bond which is between two sugar units, or a sugar and a lipid, or a sugar and a protein will be catalyzed by a different enzyme. So you may recall that for glycogen synthesis, which is of course not a modified protein, but it is a polysaccharide, we needed one enzyme to bind glucose units together, and then another one just for the branch point. Now, for every different bond, we need a different enzyme. So depending what will be upregulated among the glycosyl transferases will determine what kind of glycosylation we can put on the protein. Okay? I also emphasized it before at glycogen synthesis, that every biomolecule that you want to put into a biochemical reaction needs to be activated. And we talked about activated sugars, and in case of glycogen synthesis, it was UDP glucose. Now you can imagine that every sugar, modified sugar, needs to be activated. So it can be, these are activated by nucleotide, for nucleotide sugars. So it could be UDP, it can be CNP, etc. What you need to remember, activated sugars and glycosyl transferases that need to be working to assemble the unit that you want to put on the protein and the enzyme which will actually join that carbohydrate to the protein. Now, of course, when we synthesize this very complex molecule, we need to degrade them, and the degradation is catalyzed by glycosidases. These are enzymes that are hydrolases, which means that they can hydrolyze specific bonds, specific glycosidic bonds. So as many glycosyl transferases we needed for synthesis, as many glycosidases we need to break those complex carbohydrates and the adjoinment of the carbohydrates to the proteins or lipids. Now, these glycosidases reside in the lysosome, and you will learn later about many genetic disorders that are related to the breakdown of these modified proteins, oligosaccharide units, or polysaccharide units. So, we put this modified protein bicarbohydrate into two different groups. One we call glycoproteins, and the other we call proteoglycans. Glycoproteins that are glycosylated by either monosaccharides, unmodified or modified sugars, and oxidized sugars. They can be either monosaccharides or oligosaccharides, can be the same, can be different components of that. And usually they are branched, which means that not just the long array of sugars which we produce, but there will be branch points there. Proteoglycans are different. There is also a core protein that you need to glycosylate, but this glycosylation is not by mono or oligosaccharides, but by so-called glycosaminoglycans or GAGs. These GAGs are composed of repeating disaccharide units, and they are further modified with sulfation. They are never branched. So what do I want you to remember? Glycoproteins, monosaccharides, or oligosaccharides. Not really the very, very, very long polysaccharides that will decorate the surface of the protein. It can be simple sugar, modified sugar, oxidized sugar like uronic acid, and they are branched. The proteoglycans are proteins that are modified by glycosaminoglycans, GAGs, which contains repeating disaccharide units, modification with sulfation, and they are not branched. Okay? To remember better, I put here two figures for you. So this is a typical picture of a glycoprotein. This is a typical picture of a proteoglycan. This is the proteoglycan here. What is common? We have a core protein here. We have a core protein here. Don't bother about G1, G2, G3, and so on as of now. Here are the branched oligosaccharides that decorate the surface of this protein. And here are the long chains of gags that decorate the surface of the proteoglycans. Now, where do you see them? I will talk about it a little bit later, but you see, if you look at a general cell surface membrane, can you see this dot, dot, dot structure here? Some of them, and this is actually a lipid, some of them are branched. These are those glycoproteins that sit in the cell membrane. Of course, they can sit somewhere else too, but you will actually see a lot of glycosylated proteins on the cell surfaces. And this is a schematic representation of the structure of an extracellular matrix. And what you see here is, you remember from the previous case that we talked about the striated structure of collagen, how you see the collagen. So these are big collagen fibers. And in the extracellular matrix, you have these proteoglycans also. Now, these little things there are proteoglycan. I mean, sorry, not little things, but little things here are glycosaminoglycans, and here is one proteoglycan molecule. Here is the other one. Let's start with glycoproteins. So where do you find glycoproteins? What is the function of this group of modified proteins? They usually play a role in lubrication, and they protect the tissue surfaces. For example, tissue lining of the body ducts, there are a lot of glycoproteins covering that surface. They also can be hormones. Our hormones can be modified. Protein hormones or peptide hormones can be modified by glycosylation. Our plasma proteins are heavily modified by glycosylation. And as I mentioned, a lot of cell surface glycoproteins are there for the function of cell-cell adhesion, cell recognition, and other different functions. For example, binding. A receptor is modified, and this modification is helping the binding of the specific molecule that the receptor is produced for. Now, how much carbohydrate is on the surface of the protein is variable. So, for example, the IgG, which is also modified with simple sugars, contains about 4% of its weight as carbohydrate, and glycophorin, a cell membrane glycoprotein, contains as much as 60% of its weight in carbohydrates. So why do we need this glycosylation? You already saw that a lot of glycoproteins have specific jobs. So in the previous times, we thought that, okay, it makes proteins decorated, more unique. Yeah, they are more unique so much that they play crucial roles in some biological processes. And I just put here that fertilization of the egg requires glycosylation of the surface for the sperm to be able to fertilize the egg. When we have cell death, then certain protein glycosylation units are recognized, and so on. And the cell surface oligosaccharides, as I mentioned, is very rich in playing roles in lots of things that the cell is doing. It's very important to actually change the pattern in development, differentiation, etc. Now, very important to know ahead of time that if this glycosylation is not going well, then it will give an opportunity for the immune system to recognize that surface So autoimmune reactions can be upregulated if glycosylation is not proper. And also aberrant glycosylation can start up some abnormal processes. So there was some of this related conditions described in cancer. I want to remind you to one very important um, glycosylation and difference of proteins that you already learned in the HDHR block. And these are the so-called blood group substances. The blood group substances are nothing else but decorating the surface of the erythrocyte, the red blood cells, with specific antigen determinants, with specific, actually, glycosylation patterns. And you may recall that you learned that there are about 100 blood group determinants belonging to 21 different categories, etc. So how is it that the different red blood cells in different individuals can be different, can be different from each other, that different transferases are upregulated in that particular person's body? Now, aberrant glycosylation, you remember blood transfusion? If you add blood to a patient which is foreign to the patient, then the immune system can recognize this transfused blood. It can play also a role in after birth, the mother can develop antibodies against those surface determinants that are coming from the fetal blood, and then there will be a problem, rejection of the second pregnancy. It is actually sometimes good that this glycosylation pattern can be analyzed. And this is how we analyze the blood erythrocytes for patients to put them into O or A or B or AB or Rh positive, Rh negative, and so on. A lot of different categories. It is also important that in certain cancers, this glycosylation pattern is changing, and we can detect it and it can be served as a marker. So I just put here one that PSA, prostate-specific antigen, starts to be synthesized by prostate cancer cells, a different glycosylation pattern, and we can detect it, and it's very important for early detection of prostate cancer. Now, again, it is just part of the reminder, and you learned about this all. So I just wanted to drive your attention to the O category has a specific tetramer of carbohydrates. And when we go for A or B or AB, also you can see that just the fact of one additional sugar residue or then other additional sugar residue will strictly distinguish between O or A or B. And when you have the phenotype of AB, you have this atypical and B-typical, just one single carbohydrate unit added to this core tetramer, which will generate immune response. Now, and I just wanted to remind you of this one. I am actually an OO. My phenotype is OO, so I cannot get blood from anybody but another O, but I can give blood to everybody. Good for me. All right. Now this is for your information, but I like to put some thoughts into your head. It's very important for biopharmaceuticals also to take into consideration what the glycosylation pattern of that protein or lipid, what they are producing is. Otherwise, that particular pharmaceutical product can be not useful or actually can be harmful to the human body. So one point is that when vaccines are developed, and you also learned about it in the very first block, it's very important to actually mimic very strictly that particular bacterial or viral surface glycosylation which you want the vaccine to develop for. If it is just slightly different, this vaccine will not be efficient. Now, you also learn that we try to replace our proteins, a lot of them, with recombinant proteins. So just a couple of them, insulin, albumin, blood clotting factors, now are available to give to your patients and not relying on blood transfusion from another patient, which always bring this danger with it that the patients, your own patient's body will recognize that particular foreign blood, not mentioning some infections that can be given through the blood transfusion. Of course, blood transfusion is still very much used, and very, very serious testing is going on the donor blood before it can be given to the patient. Nevertheless, so recombinant protein, insulin, albumin, blood clotting, etc., they need to mimic the glycosylation of the human albumin. So, for example, when insulin in diabetes, insulin was first used, it was porcine. It was coming from pigs, and some of the patients reacted to that one because the glycosylation pattern of the pig insulin triggered an immune response in the patients. Now, the recombinant insulin is strictly following the pattern of the human insulin, as well as the blood clotting factors. If you remember, we have a lot of blood clotting factors, and some disorders are related to the lack of blood clotting factors, so the patients need to get those. All right, so as I said, serious illness or inefficiency or efficacy, loss of efficacy, or serious illness can be developed. Anaphylactic shock also can develop because of autoimmune response or immune response, I would say. Now, drugs. Cetuximab, I don't know whether you have not learned about it, a colorectal cancer drug. Herceptin, which is breast cancer drug, very important. And they are they are monoclonal antibodies. You remember, antibodies are glycoproteins. If these particular drugs, monoclonal antibodies, have a certain glycosylation pattern, which will be recognized by the immune system, that will just take the drug out of effect, and so on. So, as I said again, it is just some extra thought into your head. When you go further in your studies, you can potentially remember some of this, how glycosylation can make a huge difference. So, how do we synthesize these glycoproteins? So, we have the protein coming off the ribosomes, and there are some specific amino acid residues in the protein to which this glycosyl moieties can be attached. So, these glycosyl moieties can bind to so-called N-linked bond to asparagine. Asparagine has this imino group at the end of the side chain. So this is an N-linked glycosylation bond, and or can be O-linked. O-linked means that you need in the amino acid side chain a hydroxyl group. So most frequently it is a serine or a threonine. Very infrequently it is tyrosine, which gives the hydroxyl group to which the first sugar molecule can be added. So this is O-linked because of the oxygen. This is something special that I wanted to bring up because you have learned about collagens, and the collagen contains a post-transcriptionally modified hydroxylysine residue, And this hydroxyl group can enter into a reaction with a sugar, and this is also a specific O-linked glycosidic bond. So please remember, N-linked through asparagine, O-linked through serine, threonine, sometimes tyrosine, and specific O-linked through hydroxylysine, very much related to what you learned about collagen before. So how do we synthesize them? After this first bond in O-linked glycoproteins is a sequential glycosyl transferase action, which means that after the first sugar bound to that asparagine or serine or threonine, then every other sugar, like just in glycogen, will bind to that first sugar. Okay? And then there will be branch points here and there. Now, in N-glycosylation, I'm sorry, I misspoke. This is the O-linked, so it is serine, threonine, tyrosine, or hydroxylysine. However, when we have N-linked glycosylation through asparagine, the difference is that at first we synthesize the complex carbohydrate, and the last step is to bind it to asparagine. Now, this is beautiful. I cannot be amazed more than this is, but I will not hold you responsible to remember the composition of these carbohydrates. All right, so we synthesize them, now we degrade them. How do we degrade? We degrade this with glycosidases, which sit in the lysosomes. Lysosomes, the specific glycosidases are acting on complex carbohydrates, and what you see here is an N-linked oligosaccharide, and then enzymes, you see 1, 2, 3, 4, 5, 6, 7, 8 different enzymes are necessary to hydrolyze these specific bonds that are between sugar and sugar or sugar and protein. Now again, this table is just for your information only, but I put it here to show that lysosome needs to have a lot of different hydrolases to be able to digest this very complex protein sugars, oligosaccharides, to digest them down to single sugars and single amino acids. So even if just one of these many hydrolyses missing from the lysosome, we develop lysosomal storage disorders. In this case, which is related to glycoproteins, we call them oligosaccharidoses. Okay? So what do I want you to remember? Enzymes are hydrolases, which we call glycosidases. Every specific bond, you need a specific enzyme to hydrolyze that bond. They sit in the lysosome, and if even just one is missing, we develop lysosomal storage diseases, which we call oligosaccharidoses in the case of glycoproteins. Again, for your information only, this is what you see. This is the very first enzyme here. So this very first enzyme is missing, which means that everything can be degraded from this complex carbohydrate. The protein can be degraded, but this single amino acid, asparagine, will be found with binding to an acetylglucosamine. These single little things, which is a remnant in the lysosome, will accumulate in the lysosome, will distort the lysosome, will kill the cell, and will cause such very serious progressive disorders that you see a picture of in this child. Any of these enzymes are not present in the lysosome will result in devastating childhood disorders. You will learn about this in more details in the second year of your studies with us. So, proteoglycans, the second group. So you remember proteoglycans are the proteins that are substituted covalently with glycosaminoglycan or GAG chains. These GAG chains are containing repeating disaccharide units, and usually these repeating disaccharide units are either glucuronic acid or a modified sugar. Sulfation is very frequent. So these are polyanionic, very negatively charged. The GAGs are. When they bind to the core protein and they form proteoglycans, proteoglycans are known to be from their negative charges. The uronic acid has a carboxyl group, sulfate groups are giving extra negative charges. So when I will list for you the GAGs, there will be one exception, which is so-called hyaluronic acid, which is not sulfated and which is not bound to the protein. All the other GAGs are known to be bind proteins and form proteoglycans. Proteoglycans can contain one single glycosaminoglycan chain, or some contain so much that 95% of the weight of the proteoglycan is coming from GAGs. Now, what are the functions? The functions are lubrication, as it was for glycoproteins too, and they are supporting elements of pericellular or connective tissues. They stabilize and support systems. They also, because they are so negatively charged, they play a role in water maintenance, salt maintenance, and they are actually giving elasticity for the connective tissue just because they can be pushed and the water goes out, or they can be relaxed and water comes back. So a couple of them that you will learn about later, especially aggrecan, major proteoglycans, aggrecan and versican in the extracellular matrix. Syndecan CD44, you remember this? This is a proteoglycan on cell surface. Thrombomodulin are integral membrane proteoglycans. Neurocan, brevican, cerebrocan, phosphocan are nervous system proteoglycans. So they are everywhere. They really are important in function. And one of the functions in the hyaline cartilage I will talk about in a separate lecture. So what are these GAGs, glycosaminoglycans? We have 6 different classes: hyaluronic acid, chondroitin sulfate, dermatan sulfate, heparin, heparan sulfate, and keratan sulfate. I will not hold you responsible to remember the repeating disaccharides in these particular molecules, but you see the difference right away that hyaluronic acid is the only one which is not sulfated. You see the sulfate group in every other glycosaminoglycan. I will hold you responsible for hyaluronic acid and heparin. So let me go there. So hyaluronic acid, repeating disaccharide, unsulfated and not bound to protein. Actually, what hyaluronic acid does, that it's an extremely long, repeating disaccharide, polysaccharide, 10 to the 5th, 10 to the 7th Dalton, very long chain. And it actually binds other proteoglycans and they keep them together. So this is in the middle, hyaluronic acid. It is a rotary shadowing electron microscopy picture. And these are proteoglycan units that are kept in the extracellular matrix as a bunch. Very important in synovial fluid, hyaluronic acid is in vitreous humor in the umbilical cord. It gives viscosity. It gives water binding capacity. So it's very important alone or in the extracellular matrix is binding proteoglycans. Heparin. Heparin, you might heard about it because it is an anticoagulant. So heparin actually has more functions. It is working inside the mast cells, but what we know heparin more for is the anticoagulant action. And how does it work? That heparin binds to antithrombin, actually it is antithrombin 3, and antithrombin is an inhibitor of thrombin, which is a coagulation factor which actually determines how much fibrin will be produced and how quickly coagulation can happen. So if thrombin is inhibited by antithrombin, this particular inhibitory effect is actually enhanced by heparin, which means that we make sure when we do not need blood coagulation more, then we can stop it either naturally within the body, or we add as anticoagulant to patient, especially post-operatively, or patient who suffered myocardial infarction. You don't want blood clotting to happen and obstruct that uh, artery again. How do we synthesize them? You remember the O-linked oligosaccharides? We synthesized proteoglycans in the same way. Of course, we need more enzyme and more intricate things, but majorly the core protein is here, and in this case it is serine, to which the first sugar, in this case xylose, will be bound. And then everything else comes after xylose, one sugar at a time. Now, it is when the core protein is coming off the ribosome in the endoplasmic reticulum, this glycosylation is already determined and is going on, as well as sulfation. The sulfation is done by a different enzyme, of course, a sulfotransferase, and it uses 3-phosphoadenosine 5-phosphosulfate, or PAPS, which is actually the so-called active sulfate. It is formed from ATP that carries a sulfate group to biochemical reactions. Okay, so what to know about it? Sequential addition of sugar residues or modified sugar residues and sulfation to the determined serine or asparagine residue, serine, threonine, or asparagine residues, to form O-linked or N-linked bound between the protein and the GAGs. How do we degrade them? We degrade them very similarly. It is also lysosomal. These are also glycosidases and proteinases, as for oligosaccharides, but please remember that because these are sulfated, we need sulfatases also to take off the sulfate group, and we need any other modifications taken, not like the acetyl group from the modified sugars. Of course, lysosomal disorders, lysosomal storage diseases, are known when one of the enzymes that degrade proteoglycans are not expressed in the lysosome or not delivered to the lysosome, and we call them mucopolysaccharidoses. Mucopolysaccharides were the previous name of proteoglycans, and the disease is still carrying that ancient name. So mucopolysaccharidoses, lysosomal storage disease, where one of the enzymes that degrade the proteoglycans is missing. So this is, again, not to memorize, just this is just a simple example that you have here, a let's say dermatan sulfate GAG, and how many different enzymes you need to take off all the modifications, like the sulfate groups here, or digest the different glycosidic bonds between the residues. So this is what I brought here for today for you. So we talked about proteins covalently modified by carbohydrates. We talked about that it is a post-translational modification step. It happens in the endoplasmic reticulum. The glycosylation is carried out by glycosyl transferases, as many of them needs to be there, as many different glycosidic bonds we want to form in the oligosaccharides or polysaccharides or modified sugar between the modified sugar residues. Glycosylated proteins are degraded in the lysosome by glycosidase enzymes. Lysosomal storage diseases are developing if just one of these enzymes are not there, and we call them oligosaccharidosis or mucopolysaccharidosis. Glycoproteins and proteoglycans that we distinguish between the two carbohydrate modified proteins, glycoproteins that are modified with monosaccharides or oligosaccharides between, binding those sugar residues to serine or threonine, which we call O-linked, or asparagine, which is N-linked, and they are frequently branched. Proteoglycans are modified by glycosaminoglycans or GAGs that are polysaccharides which are built with repeated disaccharide units of sugars or modified sugars and frequently sulfated. Glycoproteins are mostly membrane components but can act in different other positions, like when they are functioning as working on the surface of different tissue cavities, and proteoglycans are mostly in the extracellular milieu, but we know proteoglycans also being in the cell membrane. Thank you.